Hi, I will be discussing the different stages of mitosis. So, beginning with interphase, in interphase, the cell replicates and grows its DNA and centrioles. DNA is the cell's genetic code that is the direction for everything the cell needs to do and make. The nuclear envelope houses the chromatin, which is a pool of disorganized DNA. The centrioles are organelles that assist in mitosis. The next stage is prophase. Prophase is where the chromatin condenses into chromosomes, a more compact form of DNA. The chromosomes, which are paired chromatids, are just two sets of identical DNA connected at the center by the centromere. The centrioles separate, moving to opposite sides of the cell. In between them is a spindle begins to form. A spindle is a fiber that comes from the centrioles. In prophase, the nuclear envelope also breaks down. The next stage is metaphase. In metaphase, the chromosomes line up across the center of the cell. So like the dark blue things right in here. And each chromosome is connected to a spindle fiber at its centromeres. This organization helps to ensure that in anaphase, when the chromosomes are separated, each new nucleus will receive one copy of each chromosome. After that is anaphase. The sister chromatids separate into individual chromosomes that are moved apart by the spindle fibers to opposite sides of the cell. Then there's telophase. In telophase, chromosomes gather at opposite poles of the cell, and new membranes or nuclear envelopes will form around the daughter nuclei. Chromosomes disperse and spindle fibers disperse as cytokinesis or the separating of the cell begins to occur. This is kind of like the final stage. In cytokinesis, the cell is pinched in half by a ring of protein called actin. That kind of pinches it around the center of the cell, creating two identical daughter cells, each with their own nucleus. I kind of help myself by knowing the different steps by thinking of I pick my own apples. That kind of helps me organize all the different steps. So I hope this works.